Alright guys, we'll be talking about lectures 20 and 21 today, so just not even waste time, let's get into it. So, let me move this. So lecture 20 is RNA synthesis and RNA processing, so we'll talk about transcription and the differences between transcription and translation. So transcription is a process of making RNA from the DNA template itself, and recall that DNA is double-stranded, whereas the RNA that we're going to make, in this case, is single-stranded, meaning that we're not going to replicate both of them. We're only going to take one of them, and that's going to serve as our template strand. So I just want to orientate you here in the bottom left corner is that we have our RNA transcript, which is going to match the DNA non-template strand, which is also known as the coding strand. And the coding strand is going to be exactly the same, except replace the T's with the U's, because we know that that's what happens with RNA. And then we're going to have our template strand, that's going to be the complement of our RNA transcript. So just a little basics, but also RNA is synthesized in the 5' to 3' direction, so it's not that it goes from 3' to 5', like the RNA polymerase reads it, the DNA template strand. So you can see here, it's reading from the 3' end of the template strand going to the 5', but it's synthesizing from the 5' to 3'. And remember, it always maintains that anti-parallel conformation, configuration, whatever you want to call it. And yeah. So how much of the genome is copied when we're comparing transcription and DNA replication? When we're talking about DNA replication, we're saying that the whole genome is copied when it's DNA replication. When it's transcription, it's only a particular subsection of the entire genome. And this makes sense. We don't have a protein that spans the length of our entire genome. That just doesn't make sense at all. And in terms of template, the DNA replication, we know that both strands will serve as a template because we have to end up building a two-stranded DNA duplex, whereas for transcription, only one is going to serve as the template. And then for primers, we know that DNA replication does require primers, whereas transcription can just automatically go without it because it uses RNAs. Okay, so RNA polymerase synthesizes RNA by reading the template DNA. This is very standard. It's the same way as like DNA polymerase, other than the only difference is DNA polymerase incorporates the T's, RNA polymerase incorporates the U's, the uracils. And again, RNA polymerase does not require a primer, it just keeps going, whereas DNA polymerase does need that primer in the first place. And again, it synthesizes RNA from the 5' to 3' end, and it uses ribonucleotides that have the 2' OH. Remember the difference between deoxyribose and then ribose. So just review that if you need to, but that's just one of the things that you need to know in terms of subtle differences between the two types of DNA, like replication and transcription. And here are the essential components of a gene. So specifically here, we have the coding strand and template strand, and as you can see, for the template strand, it's going to the RNA polymerase is going to bind onto here from the 3' end. And remember, it reads the template strand from 3' to 5'. But it's going to synthesize RNA from 5' to 3', and we always have to maintain the anti-parallel configuration. So the promoter is just a region of the gene that allows for the RNA polymerase to have higher affinity, and we'll talk about that with sigma factor or with the eukaryotes is something else. But these factors are going to bind onto the promoter and then allow RNA polymerase, to bind onto it, and then from there begin transcription, right? So this region is technically not going to have the transcript per se. It's going to be this region where we have transcription starting. And so transcription was going to go. There's this introns, but we'll talk about intron splicing later on. And then transcription is going to end at the terminator region where the sigma factor is going to come off, or the other transcription factors are going to basically come off, and the RNA polymerase also leaves, and then we end our our transcript here. So these are just the general regions. You should know the promoter. You should know specifically what are the exons, introns, and you don't need to know specifically for which gene to have that memorized. I'm just saying just know what these general regions mean, and then you have the terminator here as well, and then know the anti-parallelness and coding strand versus template strand. That's pretty much all you really need to know. So there are 4 phases of transcription. There is assemble, initiation, elongation, and termination. So the assembling part is just bringing the complex together and allowing for it to bind onto the promoter region. And it's initiation, right? It's what ends up happening to this complex that allows for what allows from the closed complex to go to open state, and then from there initiate transcription. So that's that step, which we'll go in depth. Then there's elongation, which I don't really need to tell you guys that in depth, other than that the RNA transcript grows longer. And then there's termination, where we reach the terminator region and the RNA polymerase is just going to come off of the transcript, or off of the DNA duplex itself. And so this looks very familiar to DNA synthesis: initiation, elongation, termination. So in terms of, we're going to talk about prokaryotes specifically. So talking about initiation and elongation, what's going to end up happening is, I know this is in the way, but For our RNA polymerase, it binds to the promoters, and it's basically these specific DNA sequences, as I mentioned before, and it's like a consensus. So you can think of it like the Tata box or something like that. I'm sure that name is familiar, but if it's not, look up Tata box. What does it mean? It's just a general promoter. But essentially, what ends up happening is the RNA polymerase and the sigma factor are going to combine together and basically attach itself to the promoter region. The way it works is normally it says preformed, right? Yeah. So the sigma factor is going to bind onto DNA, and then from there, because of that promoter region, then because it's already a preformed complex, the RNA polymerase is already going to be attached to the sigma 70 factor, and then from there the transcription bubble can form, and that's when it starts opening, and then we can have transcription proceed and enter the elongation phase from here. So right here, it binds to promoter and forms a closed complex, and then transcription bubble opens, and then they form an open, and then transcription is initiated. And then basically we have to get rid of the promoter as elongation occurs, because the sigma 70, this is like a promoter protein, whatever, for the RNA polymerase. We have to get rid of this in order for elongation to continue occurring, or else it's just going to get in the way. So that's what's going to end up happening here as the elongation is about to proceed, and once that happens, elongation just continues. And then NUSA, you don't, it's an elongation factor is what you can call it, essentially, to be more broad. You don't need to know a specific name. And then transcription terminates and NUSA dissociates, which is just, again, the elongation factors. There are two ways to terminate transcription in prokaryotes. So the first way is the row independent. So this P it's a Greek symbol for row, and then there's the row dependent. So for the row independent, it means it doesn't require any additional factors or proteins, and termination relies entirely on the specific features of the RNA transcript itself. So for example, as you can see in this diagram, there's elongation, and then we reach a terminator sequence, right? And the RNA transcript forms a hairpin loop. And this hairpin loop just basically comes off. It basically dissociates itself from the RNA polymerase, and the mRNA is released. Does that make sense? So like normally if this hairpin loop wasn't here and it was just like a straight line like this, it would just keep elongating, right? But it's the fact that the RNA transcript itself forms these favorable hydrogen bonds to form this hairpin structure that it kind of like forces itself off, like almost like ripping it apart, just because of the conformation. It can't elongate like this anymore. And so this is one way where we have mRNA that is released. But now in this other one, it's row dependent, meaning that termination requires row. And row is just like a helicase enzyme that basically attaches itself onto the RNA transcript. It uses ATP hydrolysis, makes its way towards the RNA polymerase, and then once it reaches it, then it basically detaches and kind of like cuts it, the RNA transcript from the RNA polymerase, and that's how you get rid of your RNA transcript from there. Now, in terms of eukaryotic versus prokaryotic transcription, the RNA polymerases in eukaryotes, there are multiple RNA polymerases, and they're for different types of RNA. So for example, RNA polymerase 1 is going to be for the rRNA, and you do have to know which one is specific for each function. So like take the time to get to know these, just put them through flash. RNA polymerase 2 is going to do mRNA and non-coding RNA, as you can see here on this top left diagram. And then you have RNA polymerase 3, which is tRNA, rRNA, and non-coding again, RNA. And there are many transcription factors, again, in eukaryotes, because we're just more complicated like that. There are C-terminal domains as well on eukaryotic RNA polymerases, which are just basically another way to regulate the elongation step, the transition from initiation to elongation. This is another way that we can regulate it by having phosphorylation occur or not occur. If it doesn't occur, it won't begin the elongation transition, or transition to the elongation phase. And so how many genes are transcribed in prokaryotic? I want to bring up this concept of polycistronic, which just means that genes are transcribed as operons, meaning that you only need one promoter, when you have one promoter, it transcribes multiple genes. Whereas monocistronic means that if we have one promoter, and it only transcribes one gene for that promoter. So prokaryotes, they use this polycistronic because it's much more efficient. It doesn't cost as much energy, right, to get the whole machinery, the transcription factors, the transition from initiation to elongation. That takes a lot of energy, and then on top of that, dissociation. So if you have all of this kind of combined, it makes it much more efficient. So that's why prokaryotes use this. And again, it transcribes multiple genes at once, so it's like very efficient and effective. And then eukaryotics, it's one by one by one by one. Okay, so for eukaryotic transcription, there are a lot of things here that we'll go over, like the very specific details before we go into them. We'll go over like the main idea versus going into like the small details. So just want to talk about this like top part diagram where we have factors for each step. So there's transcription factors, elongation factors, termination factors. Like you don't need to be as specific with like the TF2H and all that kind of stuff, but I'll explain very briefly. So transcription factors assemble at the promoter to help bind RNA polymerase II. RNA polymerase II, we're trying to make mRNA, remember that. So we need to have a pre-initiation initiation complex that consists of many transcription factors, so like these TFs. And the main one that you really need to know is the TF2H and the TF2E. These are the main two that you need to know, but the other ones also contribute as well. And so this is going to be a closed complex. It consists of 30 plus proteins. You don't need to know all of them, just these two general ones. And it forms a transcription bubble. What this means is kind of like a replication fork, right? Where it kind of opens up. It's the same thing here, as you can see right here. Transcription bubble forms. And now we're going from the closed complex to the open. And so for promoter clearance, TF2H unwinds DNA and phosphorylates the CTD. So these are two ways we can regulate the step from initiation to elongation of RNA polymerase II when the complex is open. And then from there, as you can see, transcription initiates by dissociation of TF2H and TF2E. So these two, that's why I'm saying they're like important. So these two dissociate, right? And now we're left with just the other TF2s, whatever they're called. We just split those, and as you can see, the CTD is also phosphorylated as well. So those are the two things that you need to know, like in terms of initiating transcription and turning it into elongation, per se. Or initiating transcription. And then with elongation, RNA polymerase II is released from the promoter and begins synthesizing the RNA transcript. And it's aided by these elongation factors to keep it bound to the DNA itself. And as you can see here, we have the elongation factors kind of inserting itself and creating more stability within the RNA polymerase to stay bound, to have that high processivity. And then from there, the RNA polymerase II is going to be the elongation factors of RNA polymerase II is going to be replaced by these termination factors, and that's what's going to allow for the RNA transcript that we have to be freely released and for this complex to dissociate now. And so we don't have that complex anymore. And then alpha amanitin just basically inhibits RNA polymerase II and transcription. Just something that you guys should know. I think this was tested my year, but the year after, I don't think it was. Okay, so we have what's it called? We have co-transcriptional processing. So there's caps, splicing, and tails. There's a bunch of like new things with being a eukaryotic cell that we do. So for the first thing is we have the 5' end caps for stability, protection, translation, enable binding. There's a lot of things, but specifically we'll talk about what this 5' cap is. It basically adds a 7-methylguanosine to the 5' end. And so the way you do this is you add a GTP. We know a GTP has the guanine. It also has the triphosphate. And so then when we first add that, we're going to add a methyl group via Adomet. Remember that enzyme adds the methylation groups. We're going to add a methyl, as you can see here. And then the cap is connected to mRNA via a 5' to 5' triphosphate linkage. So as you can see here, we have the 5' to 5' phosphate linkage. And that's what the 5' cap is. And again, the main functions are stability and protection of the mRNA transcript. We know it's super unstable for it being a one-stranded helix, I guess you could call it. And then for the 3' end poly A tails, these are also for stability as well. And this is on the 3' end, so it's on the opposite side. And usually the 3' end poly A tail is just 30 to 100 adenine nucleotides long, and we add ATP to this in order to catalyze it. And so as you can see here, polyadenylate polymerase is an enzyme that catalyzes it. You guys kind of should know the enzyme for it, and that it just uses ATP. And it adds to the string of adenylate, or the adenine nucleotides. So splicing, we're going to keep the exons and remove these introns, right? So the exons are the coding sequence, which allows for amino acids to begin attaching itself through tRNA. We'll talk about that later. And then introns, they don't have the coding sequence. There's no amino acids that can be attached to it. They're spliced out during this mRNA processing. And then there are different splicing mechanisms for mRNAs, and we'll talk about these in the next few slides. But first, before that, I want to talk about ribozymes. Very similar to enzymes. So ribozymes are RNA molecules that are catalytic, and they act as enzymes themselves. So for example, there are these group 1 introns, which use RNase P, which is just tRNA processing, and then there are common functions as well. So like they do phosphodiester bond cleavage. They also do transesterification reactions as well. But these are just like general overviews. You guys should just know generally what is a ribozyme, what are some practical examples, and we'll go through each of them here. So with group 1 splicing right here, these are in bacteria, lower eukaryotes, plants, like much more simple like organisms, and basically it's self-splicing. So as you can see here, this is a ribozyme right here. So we have our mRNA transcript, right? And we have this intron that we want to get rid of. There's a 5' splice site that begins after the U, and there's a 3' splice site that begins before the U. And so right here, what ends up happening is we have a transesterification reaction where we have this 3' OH of guanosine acts as a nucleophile and attacks the 5' site at the 5' splice site. So right here, as you can see, this is the guanine nucleotide, and the OH is going to basically attack it. The 3' OH of this guanosine is going to attack it and essentially just do like a substitution reaction, I guess you can call it, or transesterification to be more specific. Once this happens, you're going to basically have this like lariat form. Well, no, it's not lariat. You're going to have this like string of the intron. One end is going to be detached. The other end is still going to be attached. What's going to end up happening is, again, that hydroxyl group that just added itself, it's going to attack and basically replace and get rid of this intron finally. And it's going to attach the two U's together from there. And as you can see, we removed this intron from the mRNA sequence. And so from here, this intron doesn't form a lariat. A lariat is just simply, and I'll show you on this picture, this is a lariat right here where it just kind of forms a loop. It's like a lasso, essentially. So here we see that it doesn't form that lasso at all because, again, it's just like we're like ripping it apart. You can think of it like Velcro. I don't know. We're basically ripping it here and then ripping it here, but it doesn't like combine together. So that's what it means when we say there's no lariat form. There's none of this like lasso thing. And then the hydroxyl group is not within the intron as well. So this hydroxyl group is going to stay bound to the mRNA transcript that we have. So now talking about self-catalyzed intron splicing through lariat formation. So this is group 2 now. Group 2, what we're going to have isn't the intron have this internal hydroxyl group attack. So with this previous one, with group 1, we have an external hydroxyl group, not within this mRNA transcript, right? We have this one. This is an external hydroxyl group, so it'll attack and do its thing, right? Whereas this one, it's internal. So what ends up happening is we have a 2' hydroxyl group, not a 3, a 2'. This is very important. A 2' hydroxyl group on the intron is going to attack the 5' end of the intron. And when that happens, we're going to form this almost like loop, right? This lasso. And it's going to be a 2,5-phosphodiester bond, as you can see here. So it, again, forms like a loop, like a lasso in a sense. And on the 3' end that we just basically detach the intron from, it's going to, again, do the very similar thing as group 1 and attack the other side and displace this lariat. Now, this is a lariat because it looks like a lasso, essentially. And again, this step right here, this second transesterification where we have it attack here and here, looks very similar to here as well. The only difference is where does this hydroxyl group come from? Group 1, it's external, right? As you can see here, it's external. Group 2, it's internal. So just keep that subtle difference in there and know specifically which one makes a 2', or know which carbon the hydroxyl group is found. Yeah, I think that's everything for that side. Okay. So then there's also intron splicing via spliceosomes and lariat formation. So this is, in terms of lariat formation, it's going to be very similar to group 2, but it's going to do it through a different mechanism. So for the first one, we need to recognize that splicing sites, the 5' splice site, the branch point, the 3' splice site. So as you can see here, we have the 5' splicing site right here, we have the 3' right here, and the branch point is going to be indicated here. And the branch point essentially is just where the branches form. So first thing is that we need to assemble the spliceosomes. So it's U1, U2, U4, 5, 6. You can just think of this as 1 through 6, but omit the 3. And U1 pairs with the 5', and then U2 pairs with the branching point. And we'll talk about the branching point a little bit. Oh, I didn't even talk about what like spliceosomes are. So spliceosomes are complex composed small nuclear ribonucleotides. So snRNPs plus small nuclear RNAs. So that's, you can just think of it as like these U's on this diagram. But that's what spliceosomes are. They're a specific type of RNA. So we know U1 is going to pair with the 5' splice site. We know that U2 is going to pair with the branching point. And what's going to end up happening is we're going to activate the spliceosomes by releasing U1 and U4. And now we're left with U2, 5, and 6. And the first splice reaction forms a lariat, right? As you can see here. That's the first one. And it's kind of very similar to this first part where instead of having this internal, we're just using spliceosomes to catalyze this reaction first. And then the second splice site joins the 2 exons and releases the intron as a lariat. So you can think of this number 4 as specifically right here, this last step, where we have the 3' end attack the 5' end, if that makes sense. And then it basically essentially release the lariat. So in here, we have the 5' end, and I know it's not necessarily the hydroxyl group on it, I'm just giving you similarities so this is easier to comprehend, where the 5' end is going to attack the 3' and release the lariat and combine the 2 exons together. And then we basically disassemble all the proteins, and that's the entire process itself. So this is just like an intron summary. I would really go through this, the summary, the 2 transesterification reactions, conservation of phosphate bonds. The first step, attack by OH, one is from the solvent, external, the G, or one is from the intron, from the A, the adenine. And so just kind of go through this chart, understand what the subtle differences are, the nuances, and you should be good with alternative splicing and whatnot. Or with intron splicing, sorry. And then this is where I want to talk about alternative splicing. So remember how I said eukaryotes are monocistronic? So essentially for one promoter, let me show you. For the one promoter, here it is. So for the one promoter, we're going to basically produce one gene, right? Instead of these 3 that's shown here. This is a polycistronic. For the one promoter, for the eukaryotes, for the monocistronic, we're going to have one promoter, one gene. But I will say that the one gene can have different isoforms, and that's through alternative splicing, and this is intentional. So not every exon needs to be in every protein, right? So if we're talking about here, right, we have the one promoter, and we form this entire transcript, right? This has the introns, this has the introns, the poly A tail. This is all one gene. But we can take out parts of it at a different time. This is alternative splicing. So it's still monocistronic, but we can generate different proteins just because of this processing. So when we talk about monocistronic, we're talking about the transcription part, taking it from DNA to RNA. And once it's RNA, we can do a lot of things with it with processing. And so this is one of those things where we have alternative splicing. So I just really wanted to make that extremely clear. The alternative splicing is not necessarily just a eukaryotic exclusive thing, and it does not mean that just because eukaryotes can do alternative splicing that they are now polycistronic. That's just not true. They are monocistronic, but the gene is so long that we can cut it in different ways and combine them differently. That's it. But that's separate. That's processing. That is not the transcription part. Sorry for that long-winded story. And so again, different combos of exon produce different mRNAs, scripts, and proteins. And again, this is through processing, mRNA processing. So something I want to talk about is vertebrate rRNA processing. So it occurs in the nucleolus, meaning that it's inside the nucleus. And there are these things called SNO RNAs that conduct direct modulation and cleavages. So right here, just want to talk about, so here's the cytoplasm, right? And then the nucleolus. So we're starting from the top. The SNO RNAs are going to come in and essentially attach and do methylation. They can do pseudouridine formation and other modifications like glycosylation and all that. And these modifications allow for these initial cleavages, and they signal for that. There's like a specific signaling sequence. You don't need to know the specifics, but just know that SNO RNAs modify your pre-RNA or your RNA transcript for processing. So RNA splicing and processing. So take rRNA instead of mRNA, splice out the introns, and express mature RNAs to produce different size ribosomes. So as you can see here, we have our pre-rRNA transcript here. And again, we're going to do methylation, pseudouridine, dihydrouridine formation, and it's going to modify this rRNA. And then what ends up happening is we do a bunch of cleavages, we do a bunch of processing, and that allows us to develop our mature rRNA and also our mature tRNA as well. So that's just something to know about splicing and processing, but specifically for the rRNA. And then in terms of tRNA splicing and processing, for the tRNA, we have ribozyme RNase-P that makes initial, I think RNase-P, just RNase, makes initial cuts to the primary transcript at the 3' end and the 5' end. So as you can see here, the RNase-D, RNase-P are make their cuts at the 3' and 5' end, and that's going to allow for this, It's going to allow for this splicing and processing to occur. So again, we also need to splice out the intron. So that's just like processing, right? The 5', you can think of this like similar as the 5' cap and the 3' poly A tails. You can think of it similar. It's not quite though. But we also need to splice out the introns as well. but we also need to splice out the introns as well. And so as you can see here, we have our intron indicated in yellow here, and we're going to use a unique splicing mechanism that requires ATP and endonuclease, and the phosphate comes from the ATP. So as you can see here, we're going to basically cleave these, and what's going to be happening is we form our mature tRNA from there. So here is the mechanism or the process. You don't really need to know the arrow pushings per se. You just need to know that there's an intron within the cRNA, and this is linear. This one's like curved to show the hydrogen bonding and everything. We're going to use an endonuclease, take out the intron, and then from there we need to link the two exons together within this tRNA. And the way we achieve it is through this ATP route. So we activate the 5' OH using this ATP and a kinase, and then once it's activated we use a ligase and another ATP and allow this nucleophilic attack, which allows us to basically combine the two exons together. And so we form the 3,5 phosphodiester bond, and then from there we release our final phosphate group, and now it's fully attached from there. So now talking about microRNA and ribonuclease and regulating gene expression by acting on RNA. So we'll talk about these two different things. MicroRNA are 22 nucleotide long RNAs that regulate gene expression. So you can think of them as silencing RNAs that we don't want expressed. So for number one, we prepare mature mRNA using Drosha and Dicer enzymes. And so like you just need to know that specifics. You don't need to know all the specific steps or anything like that. And then once we form our mature mRNA, it binds to complementary sequence on target mRNA. So the mRNA is going to bind onto it. And then once it's bound, it's going to recruit RNA-induced silencing complexes to inhibit translation or induce RNA degradation. So mRNA attaches itself to the mRNA. So we have our transcript. It gets down to an mRNA. And then a RISC complex protein is going to form. And what ends up happening is we can either inhibit translation or we can undergo mRNA degradation. So one of the two. So that's how microRNAs work. And ribonucleases are enzymes that catalyze direct degradation of mRNA. So essentially they'll cleave the transcript itself, and then from there different RNAs are going to recognize this cleaved transcript and then promote degradation from there. And some of these are aided by exosomes in eukaryotes, which feeds the RNA to the nucleus essentially. So it's like you're shuttling it and making it easier for the RNAs to actually do its thing. And so I want to talk about reverse transcriptases. So there are some enzymes, like for example the HIV virus. It uses a reverse transcriptase to convert the RNA that it injects into DNA, and then that will be incorporated into the host genome. So specifically with reverse transcriptase, we're going to be using a template RNA to form DNA. And so as you can see here, we have dNTPs, we have RNA-directed DNA polymerase, and what's going to end up happening is we form RNA-DNA duplex from an RNA-directed DNA polymerase here. So that's what this is right here. So we form our RNA, which is in red, and the DNA is in green. And then from there, we have RNase H, which is going to basically degrade our RNA strand, and then we have now a single-stranded DNA. But what ends up happening is that DNA-directed DNA polymerase is going to come in and using new free nucleotides is going to form the double-stranded DNA. And then here's a practice question, but that's pretty much it for transcription, processing. I know it's a lot. The best way to keep it organized is like, okay, transcription is one part. Then we have the processing in the second part. But processing looks different for these different mRNAs, microRNAs. They have different functions. And so it's just good to know what each one does and then know kind of the overlaps between them, like where you see each of them playing out. Here's a couple of practice questions that you guys can do. Here's a practice question you guys can do on your own time. And then for lecture 21, translation. This one's a little bit more complex, but we'll get through it. So for translation, we have the genetic code, and one of the things is that it is redundant. And what we mean by redundant is, like, for example, here, we have UCU, UCC, UCA, and UCG. All of these are different codons, right? And before I get into it, a codon is a non-overlapping triplet code. So like any combination of bases, but it's just 3. Like that, it's units of 3 essentially. So this could either be like an ACU, it could be any of these. So that's what a codon is. And then for the 20 amino acids, we need at least 3 nucleotides per codon. And the reason why is because if it was to be 2 for the codons, well, if we take 4 to the power of 2, that's only 16 possible codes, 16 amino acids, where the other 4 we're missing that. Whereas if we have 4 to the power of 3, for the 3 codons, we now have 64 possible codons, which is just more than enough for the 20 amino acids that we have. But now you may say, well, if we have 20 and there's 64 possible, how do we mean, where are the rest of the 44, you know? or the rest of the 44 amino acids? Well, the thing is that this code is degenerate, and what that means is, for example, on this column or in this box right here, we have UCU, UCC, UCA, UCG. These are all different codons, right? They're separate, they're not overlapping or anything, but they code for the same amino acids, and this is intentional, and this has to do with the wobble hypothesis and all that. We'll talk about it soon, but I just want to say that this codon table is degenerate, meaning that a lot of them, even though they may have different unique codons, they may code for the same amino acid. So that's all I wanted to say. And then the only codons that you should be very, very familiar are the start codon and then the 3' stop. That is it. You don't need to know everything else, but the stop and start, you should just know in terms of recognizing where translation starts, where translation ends. And so like AUG, start codon, encodes for the methionine always. AUG, let me see if we can find it here. AUG, it's the start codon, as you can see in green, it's methionine, always. So every protein is going to start with methionine, essentially, once it's being translated. I know there's translation processing, which could mean that we get rid of it, but it always starts with that. Then we have UA, UGA, UAG. And the way you memorize these 3 is that you are annoying, you go away, you are gone. And this is like an MCAT mnemonic, so take that as you will. And here are the 3 in the red as well. Yeah, and as I was mentioning, there's degeneracy of codons mitigates the effect for mutations to ensure the accuracy of protein synthesis. So oftentimes when we talk about silent mutations, right, if this lets me go back. Let's say, for example, we have UCC, and let's say it mutates to UCA. Well, that's not an issue because they're the same amino acid, so it's a silent mutation. But sometimes that mutation could happen on the first one, right? So let's say we're back at UCC, right? And let's say we change it to ACC. So if we go to ACC, we go from serine to threonine. And so that is not a silent mutation anymore because we changed the protein entirely. Now, it could be like either an advantageous or a deleterious mutation, one of the two, but we won't know until we examine the protein function. So again, degeneracy in plain simple English is multiple different codons can encode the same amino acid. The only exception is methionine and tryptophan. Those are the only ones that have only one codon for that one amino acid. And it minimizes deleterious effects of mutations. So for example, UUC, it both encodes for phenylalanine, so this mutation wouldn't change the protein that you form. And the third position mutations can be silent, and as I mentioned. And again, because each codon contains 3 nucleotides, there can be 3 possible reading frames, depending on how you read it. The way you establish the reading frame is finding AUG. So it'll read it until it finds the AUG. So like, for example, here, let me see if I can find AUG. There's no AUG here. But essentially, like, this is not like a set reading frame per se. You just keep going until you find an A and then UG. Yeah. And so there are 3 possible codons that you can start with, right? And you start reading from whichever one the start is. And again, hint, hint, it always starts from AUG. And again, how you start impacts every codon that follows. The codon sequence for each reading frame is vastly different, even if just start point by just one nucleotide position. And again, the reading frame is important because, like, if you have an insertion or a deletion of one base that shifts your entire reading frame, it gives you all completely new different amino acids. It's very rare that you have a reading frame shift and it's all the same amino acids. And so again, yeah, as it states here, it's pernicious, meaning that these frame mutations shift the reading frame, throwing off all the codons that follow the point mutation. And again, frame shift mutations affect the reading frame of the coding sequence after the insertion or deletion and alter every following amino acid coded for. So as you can see here, this is just an example of what I was saying, but if you understand that concept, yeah. And here's another way to think about it with using like linguistics, for example. The cat ate the rat, and then if let's say we had an insertion of an A after the first A, and so the cat, like, I'm not going to read it out, but essentially like it's not the same sentence, even though all we did was just insert one letter. And then if we delete that one letter, as you can see, what we deleted was the T here, and we shifted the reading frame to the left a little, or not shifted, we pushed everything to the left by 1, and that completely changes the reading frame, or the way you would read the sentence. And one thing I want to note is that wherever the mutation didn't touch, that's fine. It still reads it as normal. Okay, so now moving on to tRNA. It's basically the nucleotide adapter that brings in amino acids by complementing the codon located on mRNA. So essentially we have an anticodon, meaning that's going to be complementary to the codon itself, and it's going to pair, and with this tRNA, it's going to contain an amino acid arm that basically has an amino acid attached to it. So the structure resembles a clover leaf. I would be prepared to identify and label arms of tRNA. So like, for example, the T-Cy-C arm, the extra arm, the amino acid arm, D arm. You should definitely know like where each of them are. And then wobble position and anticodon. Like, those are the main ones. And then codon recognition is done through complementary base pairing, right? So codon bases on mRNA pairs with respective anticodon bases on tRNA, and just note the polarity, right? So for our mRNA transcript, right, we have our 5' to 3' end, whereas the tRNA is going to read from 5', and if you follow this around, to 3'. So it's going to go from the mRNA transcript, we're going from left to right, and then from the anticodon, we're going from right to left in this case, and that's just because of the anti-parallelness of this. Yeah, and then another, if you just want a general rule of thumb, position 1 in the codon base pairs with position 3 in the anticodon. So now here's the wobble hypothesis. So there's greater leeway with the third position of your codon sequence. So there's 61 codons minus the 3 stop codons that encode for amino acid, but fewer unique tRNA molecules. So how does the fewer number of codons base pair with 61? Or how does the fewer number of anticodons base pair with 61? And that's where the wobble hypothesis comes in. So the first base of the anticodon wobbles and is known as the wobble base, right? So for the first base of the anticodon, let me go back so we're not confused. The first base is on the far right. It's base number 3 for the codon. So it's the last one essentially. So just keep that straight. So the first 2 positions in codon exhibit strong base pairing with the last 2 positions of the anticodon, so Watson-Crick base pairing. But oftentimes the amino acid coded for depends on the first 2 positions of the 3-letter sequence. So, for example, if we have UU for a lot of these, it's going to bind on. But sometimes, you have an instance where A or G will code for leucine, but U or C codes for phenylalanine. And so it depends. This wobble gives a little bit more leeway for one of the 4 to occur. Now, if you're unlucky and you were originally going to code for 3 U's, but then you have a wobble base that changes it to UUA, well, now you're coding for leucine because of this wobble hypothesis. Whereas if you change it from the 3 U's to UUC, it's fine. It's like a silent mutation, even though you're not mutating DNA. It's just the way the tRNAs fit onto it. Awesome. So here's the wobble hypothesis. And so something I want to know is that the third position pairs more loosely. Here are the notable changes where the I base pairs with AUC, and then there is also GU base pairing as well. Okay, and then here are the total possible base pairs for the wobble hypothesis, where we have the 5' base of the anticodon determines the number of codons recognized by a tRNA molecule. So we'll walk through this very slowly. So for the first one codon in A or C pairing, right? So the anticodon on codon number 1, or base pair number 1 on the anticodon, base pair number 3 on the codon. Here we have a C or an A, and that just means that it's the least wobble, meaning that it's the least flexible, versatile, and most specific. And so this is just Watson-Crick base pairing, so C with the G, A with the U. Now, if there are 2 codons meaning that in the G, there are 2 possible codons in the GU pairing. So if you have a U or a G, there is 2 possible things that can fit inside of it. And so this is like an intermediate amount of wobble. So for here, if we have a U, A or G can bind onto it. Remember, U is a pyrimidine. A and G are purines, so both of the purines can combine with the uracil. Now for the G, it's a purine, and either or of the pyrimidines can bind onto it, and that's just because of the extra space. Now, note that A only binds onto U specifically, and then C only binds onto G. So that's just like, these are the 4 codons that you should know if they are on the if they're on the first anticodon, right? Out of the 4 bases that you have, if they're on the first anticodon, the third, which corresponds to the third base pair of the codon. I feel like I'm saying this wrong. So these are the 4 bases that you should know that for the first base pair of the anticodon right here, it's going to be complementary to the third base pair of the codon. And so you need to know specifically what is the wobble. And if you have a I, which is just the inosine, again, inosine is like the precursor to for the purines, so the A and the G, if you guys remember to that lecture. So the inosine can bind to an A, a U, or a C, but not a G. So that's just something you should know, is that there's the most amount of wobble here, and it's the most flexible and least specific. And remember, the inosine is not on the RNA, it is on the tRNA. The tRNA may have this inosine and it allows for that extra wobble to occur. So here's some DNA mutation terminology. So we have a point mutation, we have a missense, frame shift, and nonsense. So point mutation means that we have a single base that changes. There can be 3 types. There's a substitution, there's an insertion, and there's a deletion. So substitution is we replace a single base with a different one. Insertion is we add a single base and then it does a reading frame shift. We do a deletion, which we remove a base and it does a reading frame shift as well. We have a missense mutation, which just means there's a mistake in the amino acid sequence because we change that single nucleotide, because we change that single base pair that causes it to change the identity of the tRNA that binds onto it. And again, this typically is caused by substitutions. And then we have frame shift mutations, which just means that we change the reading frame and throw off everything after the mutation. And again, this is typically caused by insertions or deletions. Then we have nonsense mutation, which just means that we transcribe a premature stop codon, meaning that, like, let's say our protein was supposed to be 800 amino acids long. Let's say we had a premature stop codon at 400. And so we lose out on the extra 400 amino acids that we need for that protein. So that's what a nonsense mutation means. And so it could lead to no protein being produced, but I think to be more technical, there's no functional protein being produced during translation. And then ribosomes are sites of protein synthesis and consist of 2 subunits, right? So they differ between prokaryotes and eukaryotes. You will have to know the distinction. You have to know the difference between bacterial and eukaryotic on the MCAT. It's stupid, but you guys have to know. So there are 2 subunits. There's a small and a large. And for prokaryotes, that's the 30S and the 50S, whereas eukaryotes are 40S and 60S. And the S in these indicate the sedimentation rate, and they just basically it means that it measures how fast particle of given size shape settles to the bottom of a solution after being centrifuged. And no, they're not additive. That's like probably the most annoying thing, is that 40 plus 60 does not equal 100 for sedimentation rate. It's all experimental. Okay. And so here are the 5 stages of protein synthesis in tabular form. So we have our activation of amino acids onto the tRNAs. We have our initiation. We have our elongation, termination, and ribosome signaling, and then folding and post-translational processing. So there are these 5 steps. We'll go through each one. This is just all in tabular form, telling you the essential components. So here's another diagram showing you the overview of the 5 stages. So kind of going over it briefly, we have for our first step, activation of the amino acids. Basically, our tRNA doesn't have the amino acid, but then it gets activated with one. Once it does so, then we initiate the translation. So the mRNA and the amino isolated tRNA bind to a small ribosome protein, and then the large subunit bind onto it, and that allows for initiation to fully occur and elongation to start proceeding. And then from there, we have elongation occur until we reach the stop codon. Once that happens, we have termination. And basically, like, we incorporate a water molecule that hydrolyzes our peptide from the tRNAs, and now we have our protein that is formed. Our RNAs dissociate, and they're recycled, essentially, and now we're back to our usual mRNA transcript. And also note that antibiotics and toxins inhibit translation. So, like, for example, puramycin, ricin, tetracycline, like, those all inhibit it. So for stage 1, talking about charging the tRNA with an amino acid, so amino acid activation, it's 2 steps. So first thing we need to do is activate our amino acid with ATP to form an amino acyl AMP, which, as you can see here, it's the amino acyl. So here's our peptide, and then it's combined to the phosphate group, the monophosphate, and then the adenosine. Then this molecule, this intermediate, is going to combine with tRNA to form our amino acylated tRNA, so tRNA with protein. And so some of the nomenclature is that ser-tRNA ser is charged, whereas tRNA ser is uncharged. And this exponent, it essentially just this superscript just means that this is a tRNA specifically for serine, but it doesn't have the serine actually attached to it yet. It is not amino acylated. So just know this new nomenclature for this. So the way that this happens, a little subtle thing that you have to note is that the enzymes that catalyze the activation of tRNA with the amino acid, these tRNA synthetases here, there are 2 different classes that attach the amino acid in a different way. So again, you don't need to know this table, you don't need to have this memorized. This is, I didn't even know that there was a table for this before reading the slide. So again, There are two classes of amino-acyl tRNA synthetases. You don't need to know which specific one. You just need to know that they act differently in the way that they add the amino acid to the tRNA. So, as you can see here, activation of tRNA. So the amino acid is activated via adenylation. So right here, as you can see, the amino acid activated with adenylation, and then we form this amino-acyl AMP, and we lose the two phosphates, the pyrophosphates, and then they become inorganic, as we know, and that drives the reaction forward. So from this intermediate, we can either go through the class 1 route or the class 2. And specifically, we'll just go through class 1. We have our, we basically have, we treat the 3' end of the tRNA as our nucleophile, and that'll attach the amino acid to the tRNA. Or for class 2, we use the 3' end hydroxyl. Again, this 3' end of the tRNA, 3' is going to attach itself to the amino acid, and then from there, attach. So these are the two differences, and basically how to connect the two is this transesterification reaction where we switch the amino acid. So it's not, if we go through the class 1 route, we know that the amino acid is going to get attached on the 2' end of the hydroxyl, and so for the transesterification, we're just going to have the 3' end grab the amino acid, and then basically the amino acid is going to detach from the 2' end. And so this is going to be our final product for both routes. They have a different route to get there. This is our final product. And then we have prokaryotic versus eukaryotic translation. So prokaryotic, we have this the Larno sequence, which basically acts as our promoter for translation to occur, for initiation to occur. And so again, polycistronic, we translate multiple genes at once with the operons, and what this allows us to do is during translation, we can also form multiple proteins at the same time. As you can see here, these are all different proteins. And co-transcriptional, transcription, and translation occur simultaneously with prokaryotes, because again, they're not separated by compartment at all. So for here, as you can see, we have our mRNA being created, and at the same time with this mRNA transcript that's being formed, we have our translation occurring as well. And as you can see, the mRNA transcript is being formed as the RNA polymerase goes through, and we're also we're doing translation here and basically converting our mRNA transcripts to proteins. Now for eukaryotes, we start from the 5' cap to start AUG. But remember, this is not co-transcriptional because we have to take our RNA transcript and move it to the cytoplasm. So remember that RNA transcript is made in the nucleus, but we have to move it through the nuclear pores from the nucleolus into the cytoplasm. So that's why it's not co-transcriptional. And we know that it's monocistronic, meaning that the one mRNA transcript that's exported out will only encode for the one protein. And yes, there's alternative splicing, so like this is to say that the RNA transcript could be combined in different ways to form that one specific protein, right? But you're still making one at the end of the day. So moving on to stage 2, there are 3 initiation steps in prokaryotes. So the first step is a small subunit is going to combine with IF1, IF3, and mRNA, and that's going to basically allow for these translation factors to come in and bind onto the start codon. Next, what's going to end up happening is IF2 with GTP, like combined with GTP, it's going to be attached, as you can see here, binds and recruits FMET, tRNA FMET. And remember, AUG is going to code for that start codon, and it's exactly this specific tRNA that's much more energetic that gets this whole thing rolling, essentially. So this is like a very special tRNA. And then from there, the 50S subunit is going to bind. So like we make the complex first, and then the 50S subunit combines and says, okay, let's get the ball rolling. And then from there, the IF2, which kind of prevents, translation from switching from initiation to elongation, it's going to hydrolyze it, and then from there we can start the initiation phase. And then in terms of the rest of these initiation factors, you don't really need to know the rest of them. You just need to know you just really need to know the IF2 hydrolyzes the GTP and the IFs leave. That's pretty much it that you need to know. And so just before we move on to the prokaryotes, I want to talk about initiation in eukaryotes. And so it's essentially the same outcome, but the general steps as eukaryotes, but it's much more different features. There's different complexes and different things. No, you don't have to memorize everything. You just need to know kind of the more general rules. So you have multiple eukaryotic initiation factors here. That's why they're called EIFs. And basically, these join the small subunit of the rRNA, or the R ribosome, or whatever it's called. These are going to join the small subunit of the rRNA. And so these eukaryotic initiation factors are going to bind onto it. And then from there, we're going to add the GTP-bound initiation factor. That's eukaryotic-wise. And it's also going to contain the aminoacyl tRNA with the start codon. So the methionine tRNA is going to be bound also with it. And then from there, what's going to end up happening is we have our basically this complex forms, and we're going to scan the mRNA from 5' to the 3' to find our start codon, right? So it starts from the 5' cap, and once it goes from the 5' cap and then finds that AUG, then it again. Number 5, a bunch of shit leaves and large subunit joins and GTP is hydrolyzed. So essentially all our initiation factors, they dissociate, and our large ribosome, the large rRNA, binds on, and that's when it allows for elongation to now occur. But yeah, I know that there's a lot of initiation factors here. You don't need to memorize all of them. You just need to know that a complex forms, our tRNA, the start codon tRNA, binds onto this complex. It binds onto the 5' cap, scans, finds the AUG. Once it finds it, all the shit leaves, and then we have our large ribosome that comes in, or large rRNA that comes in, and then gets the whole ball rolling from there. So I want to talk about elongation. It's a 3-step process for every codon. So we have 3 RNA binding sites, A, P, and the one thing I want to mention is that E is our exit. So any tRNAs that are in the E spot are going to leave. So we'll go through this. So step 0, start codon begins in the P site. It doesn't go through the A first. It starts at the P. And so for the first step, we have tRNA binding, right? So our first step happens, tRNA binds into this, and it's going to be already activated. Okay, I see. So for this first part, right, tRNA binds onto the A site, and it's going to have an elongation factor with the GTP already bound to it. Now, the significance of this is that, as you can see here, spatially, the amino acid that's coming into this complex is facing away from our start codon amino acid. It's too far away for any condensation peptide formation to occur. So this GTP is going to hydrolyze, and it's going to shift that tRNA to basically like twist and allow for this amino acid to be right there next to the methionine. And once it's right there, we can begin step 2, which is the peptide bond formation. And so the lone pair on the end term of the A site, so right as you can see here, the lone pair is going to attack the carboxyl end of our methionine amino acid. And so then from there, the peptide chain is now attached to the tRNA in the A site. As you can see here, this is the A site tRNA, and this is the P site, the methionine tRNA. So the growing chain is now on the A site. And then from there, for number 3, we have translocation. So essentially, we have this elongation factor, GTP, enter the A site and kind of force everything to move along, right? So it shifts each tRNA over one spot, and GTP is hydrolyzed in this process as well. So as you can see here, GTP will be hydrolyzed, and that moves everything one over. Now remember, E is for the exit, so the tRNA that was for our methionine is going to leave. And our growing chain is now on the P site. We have our methionine sticking out, and then the amino acid, the new amino acid being closest to the tRNA, which makes sense. It's bound to the tRNA first in order to be activated. And so then that's elongation. And then we just repeat this process over and over. Once it's in the E site, this one just dissociates, this old tRNA that's already used up. And then stage 4 is termination of translation once you hit a stop codon. So for example, let's say we hit a stop codon. Instead of another tRNA binding onto it, a release factor is going to bind onto that. And when a release factor binds, it's going to hydrolyze the peptide tRNA link. And as you can see, it's hydrolyzed and forms this free carboxyl group. And so that's why we say proteins are synthesized from the N to C terminus, because it ends off with the C terminus here. I don't know if that was convincing or not. But I wanted to mention that once this release factor binds, it's going to dissociate this entire complex, and from there we now have this mRNA transcript that no longer has any of these complexes. Translation's pretty much over at this point, and so that's basically termination. Like, those are just the main important things that you have to know. And so stages 1 and 4 are very expensive. So we have 2 ATP equivalents for just the amino acylation. So activating our tRNA with the amino acid takes 2 equivalents. Then we have our GTP hydrolyzing to GDP for proofreading. We also have elongation. So every amino acid that's added, we have to hydrolyze. And then GTP is hydrolyzed into GDP for translocation, moving it from the A site to the P and from the P site to the E. So in total, 4 NTPs are used per peptide bond, and 1 GTP is used for initiation. And then stage 5 is protein processing. When protein processing and then folding it to the final shape. So we can have a lot of these things. You should just know generally what they are. You don't need to like know everything verbatim, but what I'm trying to, you just need to know specifically what each thing means. So for example, glycosylation, we're attaching a carbohydrate side chain to form glycoproteins. Isoprenylation, we attach a thioester or thioether linkage to a cysteine residue. Addition of a prosthetic group, biotin, heme, hemoglobin, that kind of stuff. Proteolytic processing, so trypsinogen, trypsin. So for example, protein could be proteolytic, meaning that, or sorry, it could be a zymogen, and we know that once it's cleaved, it turns into an enzyme. So that's an example here. We have a disulfide bond formation, which forms cysteine-cysteine bonds. We also have amino acid modification. We can phosphorylate, carboxylate, methylate. We can have a loss of signal sequencing, which is by signal peptidases from the ER, the endoplasmic reticulum. We have the N and C terminal modification, which is just removal of the formal group. Again, the point that I'm trying to stress here is that proteins can also undergo processing just before they're used for their function. And again, we can also add a lipid chain to tether or localize proteins to a membrane as well, like RAS here. And yeah, I think that's all the extra stuff here. And signal sequences of proteins direct their localization to different parts of the cell. Like I mentioned, if you have some signal sequences, so different bacterial proteins have different amino acid sequences that direct them to their intended locations. For example, the inner membrane, the periplasm, the endoplasmic reticulum, vesicles, endosomes. Like, in order to direct where these proteins are going to go, they need to have signal sequences, and that's inherent with the amino acids that are contained within this peptide chain, right? So, for example, for inner membrane proteins, we may have a lot more lysines in it, whereas periplasmic, we have less of lysines, or they're more dispersed. They're not really grouped together, as we see here. There's a bunch of different ways. You don't need to know specifics. You just need to know that this is, again, this is a bacteria slide, but also, secondly, is that some of these signal sequences allow for these proteins to cross membranes or to stay attached to them. So nuclear localization signals direct proteins to the nucleus. Again, they're nuclear localized signals. So a protein may have a nuclear protein may have a NLS here, and that'll allow it to go through this like transport process with RAN, GTP, and all that. Yeah, NLS is on the protein is colored in red here, so it's going to bind onto this alpha beta importance will bind onto NLS and then transfer the nuclear protein into the nucleus. You're seeing this RAN GTP pathway. And so then we transfer these shuttle proteins back outside using RAN GTP, and that's going to hydrolyze to power it through. And then from there, we have our nuclear proteins inside the nucleus. That's all this slide is showing. You don't really need to know all the specific factors or proteins. You don't need to know most of it. You just need to know that there's this nuclear transport mechanism. So some proteins also may have an ER signal sequence, which means that they will bind onto the ER membrane and then either be shuttled into the nucleus, into the ER lumen, or whatnot. So a ribosome plus mRNA complex associates with SRP. So as you can see here, if we move here, the SRP receptor, or SRP, where is it? The SRP here is going to associate itself to the rRNA, and then from there, it's going to attach itself to the SRP receptor. And what ends up happening is it's almost like you're locking in the RNA onto this peptide translocation complex on the ER. And when the protein is being translated, it gets directly shuttled into the ER lumen without having to do any of this extra work. So in a way, it's like you're combining translocation and translation together. And this is for ER specific, not necessarily for the nuclear one. Because we know with the nuclear, when you translate first, and then it binds onto the specific transporter, or the important, as one would say. Yep. And then protein degradation. So there's this CLP system, which degrades proteins in bacteria, mitochondria, and chloroplasts. And so you can think of it like a paper shredder, right? We have a protein, it has a tag onto it, which allows it to bind onto the CLP. It gets funneled through, and basically we power it using ATP, and we hydrolyze two ATP molecules and break down our protein. That's all what this slide is saying, basically. And then protein degradation, eukaryotes, we have this ubiquitin protein system where ubiquitin is going to attach itself onto the target protein, and it's going to be a signal for saying, hey, cell, we need to degrade this protein because it's not useful. So that's what the protein degradation, that's what the ubiquitin protease system is. It tags the proteins for degradation with the ubiquitin as their signal molecule. And so E1 and E2 prepare the ubiquitinate protein. So as you can see here, E1 and E2 basically prepare a ubiquitinate protein, or enzyme, sorry. So right here we have our enzyme with the ubiquitin attached to it, and then from there we're going to basically transfer the ubiquitin onto our target protein, which is shown here. And then E2 and E3 confer protein specificity. These are very general enzyme names, just to say that, like, first thing we have to do is just prep it. The second thing is we have to find our target. That's all that's saying. And then we have the 26S proteasome, which degrades ubiquitinated proteins, and that's what this thing is right here. I'm pretty sure, if I'm not mistaken. Yeah, but a proteasome is going to basically recognize the ubiquitin signaling and then degrade the protein from there. And here's a practice question with acidic acid and another one. And that's pretty much it, guys. Hopefully you guys found this longer lecture helpful, but it's less about memorizing the details like EF2, EF3, TF2H, like it's nothing like that. You just need to know specifically what generally happens in the steps, which ones use ATP or not use ATP, and I mean, for the most part, we're synthesizing things, so we will be using ATP, just general concept. But yeah, hopefully you guys found this one at least informative.